Section 16.5, we're going to talk about the type 4 hypersensitivities. These are the cell-mediated or delayed reactions. So the one thing that's similar to the type 1 hypersensitivities is that it does require a sensitizing, and then you get the provocative dose. The destructive type of symptoms come the second time you've been exposed to it. Type 1, though, requires or has IgE antibodies and mast cells, where this is the cell-mediated immune or delayed reactions. And the type of cells are T cells. So the type 4 hypersensitivities involve T cells, and they result when T cells respond to antigens displayed on self tissues or transplanted foreign cells. So traditionally, this is known as delayed hypersensitivity, and the reason is because it's usually 1 to several days after the second contact with the antigen when you get the symptoms. Delayed hypersensitivity can be used in the testing in vivo testing for tuberculosis, because when you inject a little bit of the antigen for tuberculosis under the skin, you get a reaction within 24 to 48 hours. So it can be used, it's what we call an infection allergy reaction. Contact dermatitis is the more common example of delayed hypersensitivity. This is when you have resins from poison ivy or poison oak, or haptens in household or personal items like jewelry, cosmetics, elasticized undergarments, and certain drugs like penicillin. And it requires a sensitizing dose. Well, actually, penicillin's a type 1, but certain drugs, and it requires a sensitizing dose and a provocative dose. So the first thing that happens is the antigen comes into contact. You get these antigen-presenting cells that present to a T helper, which happens to produce tumor necrosis factor and interferon, which makes these macrophages angry macrophages. We also, the T helper will recruit T cytotoxic cells, and the T cytotoxic cells will kill the cells, the skin cells, and the macrophages will induce this inflammatory response. So you get these blisters, and this is what we call an induration is what is produced when you have this type 4 delayed hypersensitivity reaction. So of course, now we have an animation.
Harmful effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called TD cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. A contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. During contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. The carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T-cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. When the host comes in contact with the allergen a second time, the Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
So in that animation, we were looking at what happens with contact dermatitis, which is a very common type of delayed hypersensitivity. But a type 4 reaction is also a graft rejection, and this is when your immune system is looking for a particular MHC or HLA cell surface marker. And if you have siblings and parents, there's very high similarity between the close relations, but the more distant the relation, the less likely the MHC genes and markers will be similar. So when donor tissue displays the surface molecules of a different MHC class, then the T cells of the recipient will recognize that it's foreign and react against it. That's what it's supposed to do. The problem is we don't want it to reject the graft. So there are two different types of type 4. There's the graft versus host versus graft, which is when your immune system sees that it's from someone else. It's a grafted tissue, has a different MHC marker. And your T helper cells will activate the macrophages, and this T cytotoxic will lyse the cells of the graft, but both will cause the reaction. This is host versus graft. Graft versus host is when you have these passenger lymphocytes from the graft that get into the body, and they're looking for a particular MHC complex, which the recipient of this graft does not have. And so it's the actual graft is going to damage the host. Different types of grafts are autografts, which where you have a tissue that's transplanted from one site on an individual to another site, like the great saphenous vein is used in bypass surgery, and so they'll put that on the heart so that you can bypass particular parts of the heart where the tissue is dead. Isograft is from an identical twin. Allografts are from genetically different individuals, but belonging to the same species. In contrast, the xenograft is when you have different species. So it's a tissue exchange like a monkey or bonobos ape or something very close, similar. A pig valve has been used for xenograft. That's very common. But it's from a different species, and for whatever reason, it does not cause a reaction or very little reaction. So there are lots of different types of transplants. There are live donors, there's people who've recently deceased, so a cadaver donor, and then fetal tissue can be used as a donor. And they can transplant skin, liver, hair, kidney, coronary artery, cornea, bone marrow. With the live donors, you can get rid of one of your kidneys and your other kidney will take up the slack, and you'll be able to filter your blood just fine. Skin donors, that's not usually an issue because your skin will grow back. Liver, same thing. Your liver, if you take a section of the liver, it will grow back. Bone marrow, they'll grow back. So you can get those from live donors. But these other things like a heart, you can't do without heart, and you can't do without cornea. But you can give those up if a person is recently deceased. They can donate their organs, and that can be a gift of life. So the concept check for section 5, the type 4 hypersensitivities are the result of the action of what type of cell? And that is a T cell. So it's a cell mediated, and it's an overreaction of the cell mediated immunity. So T cells. Question 2. A positive tuberculin skin test is an example of what type of allergy. It's a type 4 hypersensitivity. So I don't know that I would call it an allergy, but a type 4 hypersensitivity. And exposure to poison ivy can result in blank blank. 2 terms. So this is the contact dermatitis. So it comes into contact with an allergen, a resin that's on the plant, and that causes a sensitizing dose, and when you're exposed to it again, then you get the symptoms of the poison ivy. So that's contact dermatitis, as opposed to atopic dermatitis, which is like eczema, is a type 1. The difference would be, again, T cells with this type, where it's antibody mediated in the type 1. Type 1 is immediate, where this is a delayed response. A blank is a type of graft from an identical twin, so that would be an isograft. And question 5, implantation of a swine heart valve is an example of what kind of graft? So if you get a pig heart valve, that would be a xenograft. So that's the end of section 5.